Kan du fortelle, du gikk på det et år der, og så var alt plakket borte, eller hvordan?
Ja, etter et år så heter jeg det.
Ja, for du har gått på sånn her, ja, du har gått på maks dose i, hva da, 10 år, var det?
Ja.
Ja, så det var ja, for det var ingenting som hjalp.
Nei, faktisk 15 år.
Ja, fortell videre da, altså. Jeg fortell bare kort den historien selv der, så får jeg høre. Jeg forteller nå bare kort resten av historien, så får jeg høre.
Nei, det var bare at doktoren sa at jeg aldri kunne gå og ta, og at jeg måtte stå på den med hele tiden.
Ja.
Og etter at jeg fikk olmetec, så ble jeg kjempebra.
Ja.
Men jeg var overrasket over egentlig hvor snart det ble bedre.
Ja, fortell da. Forklar meg nå, jeg får høre. Jeg forklarer mer i detaljer, sånn at andre kan forstå. Altså.
Ja, du kan se at jeg hadde sikkert hatt puste.
Ja. Ja.
Og det ble veldig fort sliten med å dra til bakken.
Ja.
Jeg vet at jeg har takket forsvant i blodårene, og det er mye lettere å puste, og det er mye mindre sliten, og det føler meg liksom som en ungdom, ja.
Ja.
Det er det jeg vet.
Ja, for jeg vet han fortalte, broderen for mange år siden, han var inne til en optiker, han skulle få sjekket synet for å få eventuelt korrigert noen brillglass, og så sa han at jeg ser at du har en immunsykdom, for jeg ser det når jeg ser på øynene dine med det her optikkgren vi har, at du har avlæring i årene, og da vet jeg at siden det der er en immunsykdom, så den avlæringen, i stedet blodårene i øynene, de er over hele kroppen din, sånn at hvis du reverserer det her på den olmeteken, så forsvinner alt det her, for det er over alt i kroppen. Ja, for det er jo det samme med de som går rundt med grønn stær og tror at ja, nå skal de gå til en idiot, en øyelege og skjære opp øynene så de får ut litt væske ut av øynene, men de har jo ingen hensikt, for plakket blir jo bare mer og mer. Men slår du på immunforsvaret med den olmeteken, så et kroppen opp det her plakket, så fjerner han det her han selv, og så forsvinner grønn stæren sammen med alle de andre immunsykdommene du har. Så. Men det var i Tromsø du tok den der røntgen med uten kontrast, de sparket det ut der med flestmelding i hånden. Ja, fortell litt av det. Ja, fortell litt kort hvordan de gjorde det. De sparket det bare ut med flestmelding i hånden, eller hvordan?
Nå er det på, det heter jo rite, og da kom jeg inn til en overlege der som ba meg først en kjempesvær blodfortynnelse.
Ja.
Så stakk han kanylen opp i lysken og innførte den helt inn mot hjertet så langt som den fant det forsvarlig. Og så hadde jeg sånn, det er jo mer sånn oppløsning i der, så det gikk opp og det kraftig som er i årene.
Ja.
Og så sa han at når her ser det fint ut, det er ikke nødvendig å legge inn hverken strømt eller noen ting i årene for å få god nok blodforsyning til hjertet.
Ja, for det var jo...
Ja, og når jeg var ferdig med så merket jeg at blodtrykket er veldig høyt.
Ja.
Og det var noe som jeg tenkte at det får en blodpropp opp gjennom foten.
Ja.
Og det var så kald og stikk foten, men du kan si at når jeg var i sykehus, så var det ingen fare, for jeg gikk faktisk over at det selv.
Ja.
Og etterpå da, så jeg hadde ingen problemer med å gå kraft eller noe.
Ja, og så merker du selv når du blir forkjørt, så blir det mye fortere frisk, og ja, alt mulig.
Ja, det der er jo virkelig merket.
Ja, forklar litt da, fortell.
Ja, jeg kan bare si en ting, at før sånn, særlig hvis jeg forkjørt på sommeren, så kunne jeg faktisk gå flere måneder før det var tjekk.
Ja.
Men nå så går jeg sjelden mer enn en uke, det er litt.
Ja, det er samme også. Jeg kanskje har feber en kveld eller to, og så er jeg litt snørrått kanskje i 6-7 dager, så er jeg over det. Så er jeg rett i 12 timers arbeid igjen. Det går det greit.
Ja.
Så...
Ja. Men, hva skal jeg si, Geir, jeg har kunnet gjøre det med sånne epikriser, så skulle jeg gjort det, men jeg kan ikke det.
Ja, det er bare synd at de blir kastet da. Ja.
great pleasure and honor to introduce to you Professor Marshall from Australia. He's an expert in autoimmunity in Australia, and he is giving us a talk about the autoimmune diseases, or autoimmune diseases succumb to a VDR nuclear receptor agonist. Professor Marshall, the floor is yours.
Thank you very much, Mr. Chairman. You know, 30 years ago, when I first started getting interested in autoimmune diseases, one of the things that really fascinated me about all the autoimmune diagnoses is how similar they are. What we've done on this chart is we've plotted comorbidities amongst the inflammatory diagnoses in a study cohort, a 7-year study that we've been running. And what I wanted to point out was you've got, for example, thyroiditis is a very common comorbidity with other diseases. And if you take diseases like Sjogren's, you'll find that Sjogren's is often associated with osteopenia, with, let me see, I'm trying to follow the yellow one, sorry, CFS, and some of the other chronic immune diseases. We did the same comorbidity analysis looking at some studies from PubMed, and the results are the same as we saw with our study, that very rarely do we find people, most of these are adults, very rarely do we find people with a single autoimmune diagnosis. It's usually a complex of chronic diseases. We have added non-autoimmune diseases. We've got asthma, anorexia, anxiety, and bipolar, and some of the other cancer, and some of the other chronic diseases that are commonly associated with autoimmune diagnoses. But when we've been exploring infection and autoimmunity in the past, we have really been crippled by the need to actually culture microbes. When we found microbes, really the only way that we could identify them was either by culturing them or by finding them under the microscope by happenstance. And what we can do now is we can actually find the DNA of the microbes. So even in single cell samples, we can analyze a single cell, and we can pull out from that single cell if there's any microbial DNA that exists within that cell. The NIH Microbiome Project was started some years ago and is yielding some very interesting results. The salivary microbiome, for example, this is the saliva of healthy adults. And this is a study that was done at Max Planck. And the salivary microbiome includes, admittedly at low concentration, but Yersinia, Porphyromonas, Neisseria, Streptococcus, known pathogens. And yet normal, healthy saliva contains the DNA of these species. You can't necessarily culture it, but the DNA is there. The species are there in some form. Man is really living in homeostasis with a microbial community that is the scope of which is just starting to dawn upon medicine. Here is, for example, the microbiome from a hip joint. And what you've got from this hip joint was primarily Lysobacter. Lysobacter is commonly found in for example, biofilms, and Proteobacterium, Methylobacterium. But look here, hydrothermal vent eubacterium, eubacteria that was found previously in hydrothermal vents at the bottom of the ocean with a higher frequency than Staphylococcus. Fascinating what the microbiome means. So what does that mean to us? Well, if we look at some of the studies which show intracellular microbial artifacts, this study from 1989 was done by Wirosco's group at Columbia University, and they used transmission electron microscopy and staining to identify communities of microbes which stained as microbes but living within the cytoplasm. Now what's fascinating about this is these are phagocytic cells. This was a lymphocyte taken from the eye of a juvenile rheumatoid arthritis patient. And the Columbia studies looked at a number of other diseases as well, Crohn's and sarcoidosis. Now we've been able to use light microscopy to show that as whole blood ages, the cells that are infected, this looks like a monocyte that's infected with its nucleus. The cytoplasm of this cell is very heavily infected, and as the blood ages, as it loses nutrients, the cytoplasm bursts apart, and these really thin polymers, bacterial polymers, are released into the environment, into the blood environment. Well, what we did some years ago was to find out, or to figure out, that the key to persistent pathogen survival in Homo sapiens is the VDR nuclear receptor. Because in Homo sapiens, and only in Homo sapiens, not even in some of the higher primates, but only in Homo sapiens, there is one nuclear receptor, the VDR nuclear receptor, which transcribes genes for TLR2, as well as cathelicidin, and to a lesser degree beta defense and antimicrobial peptides. All of these are essential to the intracellular innate immune defenses, innate immunity. If innate immunity fails, then the cytokine cascade that will be caused by innate immunity will cause flow on to the adaptive immune system with the random generation of antibodies. Knocking out the VDR allows these pathogens to persist, but it causes human chronic disease. If we look at some of the nasties that do that, Mycobacterium tuberculosis is a persistent infection in latent TB. That knocks out the VDR. In this particular study, they found it downregulated about 3.3-fold. We now know a lot more about why MTB knocks out the VDR. It's a different protein, TACO, but it still needs to knock the VDR out. Borrelia knocks out the VDR. Borrelia spirochetes downregulate VDR nuclear receptor by 50-fold. And then Epstein-Barr virus, the virus which I think has been associated with more autoimmune disease than any other. A very interesting study just came out from Yananamandra, which showed that Epstein-Barr virus knocks out VDR as a function of cell line, and that what's particularly interesting is that 1.5 years in the lymphoblastoid cell lines, the immature cell lines, you knock down the VDR by about 15 times. That's exactly the behaviour that a persistent pathogen needs in order to ensure its persistence. The big daddy of them all, of course, is HIV. HIV, the TAC protein from HIV, binds to the human VDR nuclear receptor, and it steals this receptor to recognise its own LTR and help it express HIV RNA within the cells, within the nucleus of the cells. Now, the interesting thing about HIV is it's a very simple genome. The genome transcribes for only 17 proteins, and yet these 17 proteins have over 3,000 interactions with the human metabolome, human proteins and enzymes. Many, many human metabolites are affected by the metagenome, the persistent pathogens that live within the phagocytic cells. The sum total of all the interactions gives rise to the totality of symptoms suffered during chronic disease. For example, if a certain person has genes from bug A and genes from bug B, which are expressing proteins and other components, proteins, proteases, in the cytoplasm, that might well lead to disease X. Whereas to get disease Y, you might need genes from bug C, D, E, F, and with the absence of bug G. This is a totally new concept in medicine. Medicine still works on the postulates of Koch, which is one bug, one disease. But now we understand it's not that. The genomes interact. The genomes accumulate gradually during life, the metagenome, incrementally shutting down the innate immune system. Genes from the accumulated metagenome determine the clinical disease symptomatology. And if we take the salivary metagenome, which is much bigger than HIV, we've got about 50,000 proteins instead of 17. And so we basically end up with an imponderable complexity, which is what you see in all the autoimmune syndromes. So the catastrophic failure of the human metabolism that we see in chronic inflammatory disease, which at first glance appears to be so diverse, is actually due to a common underlying mechanism, a ubiquitous microbiota which has evolved to persist in the cytoplasm of nucleated cells by knocking out the VDR nuclear receptor. So what do we do about it? Well, obviously, we find a VDR agonist which will switch the VDR back on again. And it turns out there is a drug in the pharmacopoeia that will do that, olmesartan medoxamil, and here is how it does it. I don't have time to go into that. When you do that, over a period of about 5 years, you can reverse just about every autoimmune diagnosis. We have diagnoses here starting rheumatoid arthritis, Hashimoto's thyroiditis, uveitis, psoriasis, scleroderma, multiple sclerosis, right down to diabetes insipidus. And these are the results of our study, which was reported by my colleague Tom Perez in Porto last year. And we were basically able to show that between 18 and 53 months of therapy, 81% of the cohort experienced reduced disease and symptoms. That's what happens. Suddenly, when you give them the VDR agonist, they become sensitive to antibiotics, low-dose antibiotics again, and the disease processes start to reverse. So I'm at my 10-minute mark. I just wanted to say that we're collaborating with West China Hospital to bring our discoveries to China, and we're actively looking for any other collaborations within Asia. Thank you.